This is a quick review of genetics and DNA. So let's start with DNA. DNA is really called deoxyribonucleic acid, and it is that building block of life. It is this double helix shape, and this is where your genes are, and those genes are your traits, right? So make sure you know what it looks like, make sure you know the parts of it, and make sure you know what it does. Make sure you know it's called a double helix, and it's double stranded, and then it's made up of these things called nucleotides. And we talked a lot about nucleotides. Nucleotide is that monomer or the building block for DNA. Has three parts. It has a house or a pentose sugar, has a phosphate group or what we've been calling the pool, and then it has a nitrogenous base or that basement. So a house, a pool, and a basement, or a pentose sugar, phosphate group, and nitrogenous base. The most important part of these three parts is this nitrogenous base. You're going to have a lot of questions on your test over DNA and genetics, and if one of the answer choices is related to the nitrogenous base, or more specifically the order of those bases or the sequence of those bases, that's going to be the right answer, because it is the order of these bases that determine our traits and that make us different from other living things and even from our brothers and our sisters. What our nitrogenous bases are, they are those letters that pair with each other. So the letters A, T, C, and G are adenine, thymine, cytosine, and guanine. So the order of these letters are what determine the RNA that's made, and that RNA determines the protein that's made. So C always pairs with G, T always pairs with A, G to C, A to T. But this order right here, C, T, G, A, C, A, that's going to determine what the protein looks like, and that will determine what the gene looks like. So here's that DNA base pairing just to make sure you got it. A always pairs with T. So anytime I have a T or an A, I'm going to have an A or a T on the other side of that DNA. And C always pairs with G. So C to G, G to C. And you're going to have to do this on your test. So they're going to give you one strand of DNA, and you're going to have to figure out what the other strand is. And then remember, this DNA is the code for RNA. And what RNA is just that code for a protein. So DNA makes RNA, RNA makes protein, and that protein is kind of what determines what that gene does. RNA is called ribonucleic acid, has the same building blocks of DNA. It has that house, the pool, and the basement. The only difference is that there is no T in RNA. So if my DNA has an A, instead of pairing with a T, it's going to pair with the letter U. So A always pairs with U, T to A, T to A to U, C to G, and G to C. So everything is the same except there is no T in RNA, there is a U instead. And that's one way you could tell the difference between DNA and RNA. So if you see a line of letters like this and it has a T in it, you should know it's DNA. And if you see a line of letters and you have a U in it, then you know it's RNA. If by chance it has a U and a T in it, then that can't possibly be the right answer because that doesn't happen. All right, so let's look a little bit deeper at DNA, because DNA is organized into something called chromosomes. So here's my DNA right here, and when I wind it up real nice and tight, I get something called a chromosome. And it's important that you remember that humans have 46 chromosomes. We get 23 chromosomes from our mom, and we have a matching 23 chromosomes from our dad. So 23 pairs or 46 total chromosomes. And on those chromosomes, there are multiple genes. So here is one chromosome, and here I have a gene, right? So here's another gene, here's another gene, here's another gene. So there's a lot of genes on each chromosome because we have thousands and thousands of genes. And what this gene is, it is a section of DNA that codes for that protein. So this part of that DNA is going to make RNA, and that RNA is going to make a protein. All right, now we could have different versions of the same gene, and that is called an allele. So remember, we get one chromosome from our mom, we get one chromosome from our dad. On that chromosome, we're going to have the same gene. So here's the gene for eye color, but we could get different versions of that gene. So maybe my mom gives me brown eyes, my dad gives me black eyes. Or hair color. Here's the gene for hair color. Mom gave us black hair, and dad gave us brown hair. Okay. And sometimes some of those genes are stronger than the other ones. So in the case of humans, dark hair, like black hair, is going to be a dominant gene or a dominant allele, and then blonde or red hair, that's going to be like a recessive allele. So here's my chromosomes. I got one from my mom, I got one from my dad, and we're going to start assigning them letters. And it doesn't matter what the letter is, just an uppercase letter is going to denote that it is a dominant gene. And then the lowercase letter will note that it is the weaker or the recessive gene. Okay. One thing that's important to remember is anytime we have a dominant version of that gene, a dominant allele, we will see whatever that trait is. So the only way that I could have the recessive version, like blonde hair, is if both parents gave me the DNA for blonde hair. And that brings up our genotype. Our genotype are the letters that we assign those genes. So remember, we have two versions of that gene. We get one from our mom and one from our dad. So our genotype are the combination of those letters. So here we have a big B and a big B. This means that we are homozygous dominant. Homo means the same. Dominant is an uppercase letter. So whatever that dominant version is, we will definitely see it here. In this case, it's a purple flower. Maybe one parent gives us a dominant version, the other parent gives us a recessive version. That is heterozygous, big B, little b. Well, that big B is stronger than little b, so I will still see the big B, so I will still have a purple flower in this case. And the recessive version of that gene, so if my mom gives us the white flower, dad gives us DNA for the white flower, that's the only way I'm going to have white flower. So there's always three possible combinations. I could have homozygous dominant, uppercase, uppercase. I could have heterozygous, uppercase, lowercase, or I could have homozygous recessive, lowercase, lowercase. And then phenotype are what the genes actually make. So our genotype was the two letters, where our phenotype is what we make. So blue eyes are recessive, so we're going to give it little b, and our phenotype would be blue eyes. So brown eyes, that's dominant, so I could have a big B, or big B, little b, but our phenotype is going to be brown eyes. All right. So homozygous versus heterozygous, I just mentioned it. Homozygous means the same. Heterozygous means different. So here I have dominant versions, big A, that is homozygous. They're the same. Big A is the same as big A. Here is homozygous recessive. Little a is the same as little a, and they're recessive. And then big A is not the same as little a, so this would be heterozygous right here. All right, now we could use that information to determine what our offspring might look like. So we're going to do that using something called a Punnett square. So in this case, I have a heterozygous mom. So here's mom's eggs. She might have this egg or that egg. Here's dad's sperm. I might pass on a big A or I might pass on a little a. So if this sperm and that egg come together, this is what my baby will look like, big A, big A. If this sperm, big A, and that egg, little a, come together, then I'll be heterozygous, big A, little a. If this egg comes together with this recessive sperm, then I'll have big A, little a. And then if I get a recessive sperm and a recessive egg that come together, I'll have little a, little a. And we could do some basic math to determine what our kids might look like. So here I'm going to have a 25% chance of being homozygous dominant, a 50% or 2 out of 4 chance that I'm heterozygous, and a 25% chance that I'm homozygous recessive. And then I could also look at the phenotypes. So I'm going to have the dominant phenotype with big A, dominant, big A, dominant, big A, little a. So it's 3 out of 4 or 75% chance I'll show that dominant trait. And then I'll have a 1 out of 4, 25% chance that I am homozygous recessive and have that recessive trait. All right, and that kind of is related to meiosis. Meiosis is how we make gametes, and gametes, if you guys remember, is the fancy way of saying our sex cell, our sperm, and our eggs. It's kind of related to mitosis. Remember, mitosis is part of that cell cycle G1S, G2M. M is mitosis, and mitosis is broken down into those 4 parts. So PMAT, or pass me a taco, prophase, metaphase, anaphase, telophase. Well, meiosis, we're going to kind of do that twice, and we're going to decrease our number of chromosomes by half. So here I have one cell. It has 4 chromosomes, one from dad, one from mom, another one from dad, another one from mom. I go through the cell cycle, I replicate them, and then I'm going to start breaking them down. Prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase, telophase 2. And now I have half of the DNA that I had in my original cell. And this is going to be what combines with the other parent's gamete or sex cell to form that new person. All right. Real quickly, let's go over replication, transcription, translation. Replication is how DNA makes a copy of itself. So here's my DNA strand, A to T, C to G, G to C, so on and so on. Well, to replicate, what I do is an enzyme comes in here and it separates my 2 sides of that DNA, and then I'm just going to base pair. I have an A here, so I'm going to add a T right here. I have a T here, I'm going to add an A right here. So I'm going to have 2 identical strands of DNA. And that DNA is going to be half old DNA, so here's my old in green, and half new DNA, which is in orange. And I just base paired it, C to G, G to C, A to T, T to A. Now I have an identical copy of my DNA. And that brings up transcription. And transcription is that fancy way of saying we turn DNA into RNA. So remember, we got to do the same thing. We're going to separate our DNA strand. Then we're going to base pair. The only difference is we're not going to have 2 strands anymore. We're going to have that single strand of RNA or messenger RNA. And there's not going to be any thiamines, no more T's. A will pair with U instead of T, but everything else is the same. Then we're going to do translation. Translation is when we take that RNA, we go to a ribosome. So here's my ribosome, and we're going to build a protein. Remember, proteins are made up of all these little things called amino acids. And I'm going to put thousands of them together, fold them up, and then that will turn into a protein, and that protein will do some specific job. And here's what that all looks like together. So here's my DNA. When I make that messenger RNA, that is called transcription. And then when I turn this RNA into a protein here, that is called translation. So here's my ribosome. All right, and let's review this real fast since some of you guys can't remember how to do it. This is that codon chart. This is how we determine what our protein looks like. And you're going to have to do this on your test. So it's going to give you maybe a section of DNA where you're going to have to turn that DNA into RNA. So if I have a T, I'm going to pair with an A. If I have an A, I'm going to pair with a U. If I have a G, I'm going to pair with a C. And this right here is called a codon, or that 3 base pairs. So A, U, C, and I'm going to use these 3 letters and this chart or the circle chart to determine what the amino acid looks like. So here's my first letter on this side. I got an A. My second letter up here is a U. So where A and U meet, this box, that is where my amino acid's going to be. And then my last letter, the third letter, is over here on the side. And I have a C. So I'm going to go over, and there is AUC right here. And that is my amino acid that I start adding to my protein. In this case, it's isoleucine. All right, I know that was pretty fast, but you guys could always slow it down, pause it, rewind it. Hope this helps for your little quiz. You could take this quiz as many times as you want. Just know you're going to have different questions each time that you take it.